All right, so I'm going to move my own head over so I can read my slides. So IPM, we're going to talk about the basics first of all. This might be review for some of you, but I think it's always important just to go over the different components. And then later when we talk about individual pests, you'll see how those components fit together in programs that we've developed in Ontario. So you can look up IPM and find a fancy definition of it, but basically what I think it comes down to is using every tool at our disposal, integrated pest management, but also considering the efficacy, the environmental impact, and economics, and not necessarily in that order either. So economics obviously plays a huge role, as does efficacy, but we don't want to ignore environmental impacts either. So I'm sure you've all seen the basic components of IPM. So we've got things like cultural control, biological control, physical or mechanical control, it's sometimes called, and chemical control. But the keystone of this is really monitoring. And I think sometimes what's forgotten in this explanation is where biological knowledge fits in. It really fills out the space in between in all of these categories, because if we don't know the biology of the pest, we don't know what it's attracted to, we don't know how big it is in terms of can it fit through screening, where does it live on the plant so we know how to hit it properly with chemical coverage, all of those kinds of things, then none of these things will work. So biological knowledge is sort of the linchpin that makes all of this function. That one of the things that goes along with that is proper pest identification, which we're going to talk about throughout this workshop. Because if you don't know what pest you have, then you can't properly, then you won't know its true biology, and you may be inaccurately applying some of the components of IPM that we're going to talk about. So like I said, biological knowledge really fits everywhere within this circle. I'm not really going to talk about economic thresholds because that gets into, lots of theory stuff, but I'm sure any of you who have studied IPM sort of know basically that it's sort of based on that you want to avoid hitting the economic injury level where your crops are damaged, so you set a threshold below that, and then you try to implement control when it hits that threshold to drive down the population, otherwise it would become too damaging. But obviously when we're dealing with flowers that are grown for their aesthetics, we sort of have to go in this with that the economic threshold is effectively zero. I mean, that's not entirely practical. We know we all sell plants with some level of pests on them, but that's sort of the headspace we're starting from. So what does that mean for the economics of IPM? I think it's important to note that so rates and number of applications are governed really by profit per pot and not efficacy. And that's what you're talking, and that's true whether you're talking about a pesticide program or a biocontrol program, because there's really not that much wiggle room between production costs and profits when you're talking about flowers, especially those grown in huge volumes. So it really comes down to what growers are willing to spend. So this is an example we have from poinsettia crops, and we work with poinsettia growers a lot on their IPM programs. And basically what it comes down to is growers are willing to spend a maximum of 10 cents per pot on their pest management programs, whatever that is. And so if we can't get it to work below that, then it's not worth doing. So you could apply all the bios in the world or do all the pesticide applications in the world, but if it doesn't get you under that 10 cents per pot, then it's not going to work out for the growers. So we've got a low threshold for pests, effectively zero, and our rates and applications and things we choose out of the toolbox are basically dictated by profit or cost per pot. So how do we manage this? This sounds like an impossible task. Well, it really comes down to thinking differently about our environment. So this is sort of the simplified model over here of IPM. We've got our different tools we can use. But this is sort of a newer model that we've started thinking about called systems-based IPM. And it's really looking at your whole greenhouse. So not only the right control mechanisms, but the right environment? Can we control the environment to make it less good for pests and better for biocontrol agents? Plants factor into that. Can we find more resistant cultivars? Can we use susceptible cultivars to our advantage? And basically, we want to think about IPM as a long-term preventative approach, understand the why behind our pest problems, what has allowed each organism to reach pest levels. That's usually because one of these things is out of balance, and address the whole production ecosystem to fix underlying weaknesses. So really thinking about IPM at each stage of production. So I'm just going to walk you through a couple examples of each of these components. So first, we're going to talk about monitoring. Mike's going to go more into this after. But essentially, it's the most important component of IPM because it really provides you with situation-specific information that you need to make decisions. And it also allows you to keep historical records, so you get a sense of what's happening in your greenhouse and what's normal for your greenhouse. So there's some tools that you can use for monitoring. Flagging tape is great if you want to flag a problem. We use white pans, or you can even use a notebook open to tap plants on, and that lets you see different stages of insects, so not just the flying insects that would get caught on monitoring cards. You can see, for example, like how much of your thrips population is in the larval stage versus adult. And then hand lenses are obviously key because they let you see your pests a little bit more close up. Other monitoring tools that are useful that you may not be aware of, spray paper. So this is a reactive paper that's yellow and it turns blue when it's hit by water. And you can affix this to different places in your crop canopy and see how your spray coverage is actually doing. So we've used this a lot with different growers who are experimenting with regular sprayers versus LVM. There's also these sifting pans that can be really useful to look at for quality control, looking at mites in sachets or even in the bulk product to see, how many mites are alive. Is there a good healthy population? Is there eggs? Are there cereal mites? So little paintbrushes are useful for that too. I think everyone should invest in a microscope. An investment of around $400 to $700, depending how much you were willing to spend. For a microscope that goes up to at least something like 40 to 60 magnification, really is so useful for seeing things like, are your nematodes alive, being able to ID different thrips species, things like that. Aspirators can be helpful too. They let you suck up pests, and then you've got live samples that you can maybe send off to your consultant or extension agent. Another tool I really like is you can get these macro lens attachments. You can just buy them on Amazon that attach to your phone camera, and you get really, really good close-up shots. So they run in the range of about $30 to $40 for a good one. You want a minimum of 15 times magnification, which would be like a hand lens. But 20 to 25 is better. But you can even, if you're feeling cheap, you can do what I do sometimes when I forget my attachment in the field, is you can actually just take your hand lens and put it in front of your iPhone or Droid camera lens and use that as a macro lens as well. So that gives you closer up pictures. So for example, like here's a picture I took of a leaf with whitefly on it, and we were able to identify that it was Bemisia whitefly versus greenhouse whitefly because we were able to zoom in close enough onto the nymphs and pupae for that. I'm not going to talk about them much because I haven't really used them, but I just wanted to make people aware of scouting apps that are out there. So these can be on your phone or a tablet. They're usually linked to the cloud to save your data. And I think these can be really good to generate reports and visualize data for multiple users. So say you're the IPM person, but you don't do the spraying, it lets you communicate, here's different areas that need attention, or here's where you should spray. It also allows you to keep digital records. So there's a couple options I've listed on the bottom. I'm sure lots of new ones will be coming out, but these are the ones I know are available now. And they all sort of have different costs and different functionalities. So that's something you can look into. So the next component of IPM we're going to talk about is cultural control. So what is cultural control? It's really just modifying the growing environment to reduce the prevalence of pests. So think about what animals need to survive. So this is my guard dog as a puppy. What does he need? Well, he needs food, lots of it. He needs shelter from the storm, and he needs the right environment. So he's partly Great Pyrenees, so he likes the snow and whatever. So if we think about that for both our pests and our natural enemies, so we want to modify the environment in a good way for our natural enemies if possible, and make the environment less good for our pests. So one cultural control that I think gets overlooked a lot is sanitation, and that's really removing the environment for pests, or the extra environment. So Mike will talk more about this later, but getting rid of all of these weeds inside and outside of the greenhouse. If you can maintain a 30-foot area of mowed lawn around your greenhouse, I know that's not always possible, but that would be the ideal. And eliminating things like pet plants, which can act as like a secondary reservoir for certain pests. You also want to promptly remove plant debris or any sick plants. If you've got a couple of dying cuttings in a liner, they're all going to be sick. So you might as well just throw them all out before it spreads to something else. Cultural control, like I said, can also be about optimizing the environment, or de-optimizing the environment is also for pests. So for example, spider mites, we know they love hot, dry conditions. So in the summer, if you increase your ventilation a little bit more to help prevent those temperatures from building up, you can prevent some of these population explosions. And I'm sure lots of you have dealt with powdery mildew, which can be the result of poor spacing or poor ventilation as well. So keeping an eye on your things like humidity, temperature, irrigation, airflow, spacing, sometimes even fertilizer. Sometimes over-fertilizing can increase your powdery mildew problems, things like that. And then in a perfect world, there goes Max. In a perfect world, we'd also be able to pick plants by cultivar resistance. Unfortunately, in the flower world, mostly flowers are bred for their beauty and their aesthetics and consumer preferences rather than disease or pest control. But there are exceptions, like for downy mildew, and that was a gigantic problem. But you can still use this, the differences between cultivars that just exist naturally to your advantage. So for example, this was a trial I did with one of my summer students years ago. We looked at a variety of spring bedding crops, and we looked at 2 to 3 varieties of each, just to look at the variation of susceptibility to thrips between all of these plants. And some of these plants we knew attracted thrips, and some we thought, you know, oh, these aren't things that growers worry about with thrips at all. So the good news is that most of the plants we tested had really low thrips numbers, so under 5 thrips per average per plant throughout the spring. But they all had standouts. So for example, there was a variety of lobularia that just was off the scale. But even crops like petunia, which you don't really think of as a thrips issue, had a variety that attracted thrips, and same with calibrachoa. So what do you do with this information? Well, what you can do is use these susceptible cultivars to your advantage. So you can either use them to guide your scouting program. So if you know a certain variety of Ribena gets hit first and early with lots of damage, start scouting there, and as soon as you see a problem there, you know to up your game with thrips everywhere else, because that can act as sort of like a spillover plant, perhaps. You can also use them, like I said, as indicator plants to know that you've got a pest problem. You can also group your susceptible varieties together, and it allows you to focus your biocontrol in one area, or even spot sprays in one area. And so one example of using them to our advantage was this work that I did with Mike Short and Rose Boutenhouse and Les Ship from Agriculture and Agri-Food Canada, where we looked at using flowering mums of a especially susceptible variety, I think it was Chesapeake, in vegetative mums. And what it did was it was able to intercept any flying thrips in the greenhouse, and they would preferentially pick the flowering plants versus the non-flowering plants. And then you could just bag the flowering plants up and throw them out. And it was a way to remove a huge amount of thrips from the greenhouse. So another component of IPM is physical control, or like I said, mechanical control. And so the biggest one we use probably in the greenhouse is, first of all, screening if possible, as well as quarantining plant material. So that I think that translates best to new plant material, especially if you're growing something you've never grown before and you may be unfamiliar with the pests that come in on it. If you can relegate it to its own little section, and sort of pay more scouting attention to that, and also just be aware of what potential issues can come in with it. That's a good idea. And then obviously sticky tape and mouse trapping, which I'm a big fan of. So like, here's an example of tape, which can be kind of unwieldy, but now they have these large mass trapping cards that are around 40 by 20 centimeters that are also really highly effective. And we use them a lot in the greenhouse industry in Ontario at a rate of around 8 per bench. And they seem to work really well to intercept, like I said, thrips coming in or that are disrupted from harvesting. They won't pull thrips out of your crop, but they'll intercept any flying thrips. So last, or second to last, we're going to talk about biocontrol real quick. So it's the control of destructive organisms using other organisms, such as natural predators, parasitoids, or pathogens. So here are some examples here. Obviously, we've got specialists like our parasitoids that will only attack 1 or 2 hosts. We've got things like generalists, some more predators, ladybugs, lacewings, things like that, things that'll eat sort of anything squishy. And then we've got our microbial pesticides, which often contain fungi or bacteria, and they tend to be pretty generalist as well, hitting a bunch of different things. And in the greenhouse industry, we use what's called augmentative biocontrol, which is basically just a fancy word of saying we dump a lot of bios in. So we release weekly or bi-weekly, and we're not really concerned about establishment. The crop's time is just too short, you know, like a 8-week turnaround for Gerbera or 12 weeks for mums. You're not going to get establishment. So it really depends on this constant supply of new natural enemies coming in. And these are all commercially reared, and they have specialized delivery systems. For example, these cards with whitefly parasitoids on them that you can hang through the greenhouse. So it's important with biocontrol to understand what it can and can't do. So what biocontrol can't do is it rarely controls an already serious infestation. So this is actually a picture I got sent from a grower recently. And he said, do you think I should use, so this is obviously spider mite, and he said, do you think I should use Persimilis on this, or is it too late? And like, once you see this number of spider mites and this amount of webbing, it's too late. So this would be a time where we would recommend pesticides. They're also not fast acting. It takes a while for the natural enemies to gain hold and gain a foothold. And we'll talk more about that in specific programs for whiteflies and thrips later when we talk about the importance of starting your bio programs early. What biocontrol can do is keep a low level of pests from increasing, which is why, again, adding them early is important. So we want to release them prophylactically rather than reactively. And again, make sure you're doing weekly or bi-weekly releases. So biocontrol also to be effective requires product quality control. So you can't just trust that what you're getting in is fine, because there's so many steps between the commercial production and the time it gets to you and then out into the greenhouse that so many things can go wrong. So first you want to talk to the producers and look at the instructions that are on the products and know how best to store them and how best to release them. But the other thing you want to do is also, there is a quality control guide that's been made by the Vineland Research Innovation Center. It's really detailed. Here's the link. I can share it with you all at the end as well. And they go through each bio and how you should test it, which I think is great. And on top of that, we've just finished some like companion videos to go along with that that are housed on Greenhouse Canada magazine's website. So if you just Google all the right ways to release biocontrol agents, you'll get our video on how to release bios, and it covers some of those methods. So the last component is cultural or chemical control, and I'm just going to quickly touch on, we know there's lots of issues with pesticides, things like worker safety, costs, resistance, and consumer perception. Obviously, those last two probably being the most important. But that doesn't mean we don't think pesticides have an important role in IPM. It's still an important component. You just have to approach it with care, and we're going to talk more on this later this morning. So that was the basics in a nutshell, keeping us 2 minutes ahead of schedule, which is great. So Mike's going to go next and share tips for successful scouting.